Hi everyone, welcome to the special webinar today. Let's wait for a minute for the people who are just joined to get together, and maybe in a minute we'll start the introduction on the talk today. And before we get into that stage, I wanna thank James again, who stayed late with us. James is from Australia, so right now in his time zone, it's actually in the night already. And for those of you who can make it, thanks you guys as well for trying to join during the day. This is like not the regular time that we usually meet up. Um, but for those who cannot meet it and are listening to the recording right now, thanks for kind of stopping by. And this is part of the 2025 webinar that will be focusing on the plant synthetic biology. And we have a lot of talks that is coming in and will be very interesting and useful for the class we're taking as well. Yeah. So before I give the floor to James, I want to do a quick introduction just to give you a brief overview of our interaction and our plan and why we are very enthusiastic to have James join us today. So let's see. I hope my screen sharing works fine. So yeah, so for today's seminar, we have Dr. James Lloyd, who's right now working at the University of Western Australia. The topic for today's talk is going to be about rewriting plant genomes for improved benefits on Earth and beyond. So that's that's probably enough to tell you what we're going to talk about. But let me give you a quick backstory about how we interact with James. So when we first posted about our plant SynBio4ALL courses that we are doing it right now, James is one of the first few person to reach to us like, hey, you're doing great. You're doing something that is like very important, and I wish that you can move forward with these projects. And at the end, we made it, and we are working on that class right now. And that's when we try to get back to James like, thanks a lot for being such a great supporter, and we would love that you share your story with us as well. So we did talk about some other plant stuff through emails, but today we're gonna learn more about what James is doing in his lab in Australia. Another thing to add on is that he trained in the UK as well before moving around. So James is a global scientist working in different setup, and he can probably add into how you guys who are also a global citizen try to be a lot of support on different continents in terms of like studying this synthetic biology as well. So with that, I want to give the floor to James and see the interesting talk for today. It's going to be around an hour of talk. So if you have any questions, please feel free to drop into the chat, and we will have some time at the end to discuss with James as well. So with that, please take it away, James.
Thank you for the introduction and the invitation. I really do think that this is a wonderful effort, a community-driven effort to take control over synthetic biology and how we can reshape life here on Earth, hopefully for the betterment of everybody. And so I really think the mission here for this organization is fantastic, something I truly support. And so, you know, most of what I'm going to be talking about today, though, will be the science. And so, but please do reach out to me, here online, a lot on LinkedIn and Blue Sky in particular. So please do reach out, follow me, send me messages. I'm happy to discuss things. Apologies if I'm a bit late getting back to you. Things are a bit hectic at the moment, but right now is an amazing time because I'm just starting up my own research group at the University of Western Australia. So for the past few years, I've been doing doing postdoc work with Ryan Lister, but now I'm continuing on with some of the synthetic biology work that started in collaboration with Ryan, and now I'm also looking into some other areas of research, linking back to some of my PhD work back from the UK as well. One thing I might draw your attention to, though, is I won't talk about it too much, but I'm part of the Australian Research Council's Centre of Excellence in Plants for Space. So this is what I consider a very interesting and unusual but fun organization to be involved with. And so this is a centre spread across different universities in Australia, University of Western Australia being one of the nodes. And the idea is, can we engineer plants to live in the extreme environments of space, so indoor farming environments, on the moon or on Mars, to hopefully fuel human space exploration. But for me, what's exciting about it is what technologies can we develop to achieve those aims that can then benefit life back here on Earth. So a lot of the synthetic biology tools that I've been developing in collaboration with others at the University of Western Australia are being used in this centre to hopefully fuel, a next generation of GM plants that could benefit both exploration of the solar system, but also benefit here on Earth. But a lot of what I'm going to be talking to you today about is the work I've done as a postdoc with Ryan, and a little bit about what I'm, moving on to as the next part of my journey as a more independent researcher. So I think I don't, this audience probably doesn't need me to lecture them about how important it is that with climate change coming, we need to be very prepared for the absolute impact it's going to have on food security. And I think we really do need to remember that great effort is going to be needed. We need to mitigate climate change, but we also need to improve crop yield. And just with traditional breeding approaches alone, I fear will not be enough to get us to where we need to be. And we have GM crops. It's evenly distributed across the world. The United States, a lot of GM crops being grown. Europe, almost none. And these are purely societal, political issues to do with this adoption. But when I think about the GM crops that are currently available and being grown, I think, well, these crops have very simple, genetic modifications. They sort of predate the coining of the phrase synthetic biology. And often it's a single gene being turned on, always continuously overexpressed. And it gives the plant resistance to a herbicide or a particular insect pest. And that's great. And we don't really see a yield penalty from overexpressing that gene. But imagine if we could overexpress something that could give you resistance to various different pathogens, bacterial pathogens, fungal pathogens. Often when you do overexpress that, including in lab models, what you end up with is a very sick plant. And so you see this plant here, my terrible cartoon drawing of a sick plant. This is an example of what you might expect if you turn on a gene continuously and it has a negative effect. It has some sort of metabolic burden on the plant. No farmer wants to grow a sickly plant like this. You could describe this as having an autoimmune disease. And believe me, I know from personal experience, having an autoimmune disease it sucks. So giving a plant an autoimmune disease where it's always thinking it's fighting a pathogen and makes it, grow very poorly, not great. So what I want to do is use new ways to control where and when a gene is turned on and off to make a healthier plant. So you can have that additional trait, whether it's pathogen resistance or production of a novel compound that is of value to you, tailoring where and when that is turned on, I think is going to be of great value. And so with the sorts of technologies I'm trying to develop, we hope to have the best of both worlds, have the benefits without the negatives. But just to really focus on, the issues of food security, we are still seeing improvements to crops, through traditional breedings. But another thing that really worries me is this is not even across the world. We do not see these improvements everywhere with every crop. And we often see it stagnating or even declining. And so yield collapses in some parts of the world, despite having new varieties being bred, this is terrible. And a lot of this is going to be purely economical and societal, cultural reasons. And, this is a major problem. But I really hope that technology, synthetic biology, for example, can help push this forward. I think we need these societal, cultural, policy, economic changes as well. But I do want to, as somebody who's not economist or a policymaker, as a scientist, I want to do my part with the tools I know how, which is play around with DNA. So the ways in which I'm playing around with DNA is thinking, well, how can you turn on a gene in a plant to make something that may help the plant grow better? But when you traditionally do this, you have a range of different promoters you can pick from. So you might think, oh, I want a strong constitutive promoter. And a lot of people who've worked with plants know the 35S promoter. It's from the cauliflower mosaic virus, strong viral promoter, has a high level of transcripts produced from it, usually leads to a high level of protein production. Is so strong and so unusual as a promoter coming from a virus, it's often a target of silencing. But if it isn't silenced, you get a lot of protein being made. But sometimes you may think, well, maybe that's too much protein. It's going to damage the plant from this metabolic burden. So maybe you choose a weak constitutive promoter, something like actin-2 from Arabidopsis. Okay, so always on, but to different levels. What about something that's only turned on in one condition? Abiotic stress, biotic stress, so from a pathogen attack, cell type specificity, or maybe you want it to be inducible only when a specific chemical is added to the plant. Well, that's great, but what happens if you want a strong promoter that's only turned on in one cell type in response to a pathogen attack? How do you discover a promoter with all those features? And if you did either discover a natural promoter like this or engineer in the lab by messing around with the promoter sequences and fusing different promoter elements to each other, maybe you would develop this. But then somebody turns around to you and says, oh, well, actually, we'd rather a weaker promoter and we want it to be chemical inducible instead. Well, then you're going to throw your hands up in the air and say, well, why did I spend all that time making this other promoter? So what I would like to see is a system where we can integrate all these signals from existing promoters that we've characterized and integrate them in to get the desired gene expression change. And so we can take something like cell type specific expression, condition specific expression, and have both at the same time without having to re-engineer the promoter, which is a huge task, almost impossible to achieve. So how do we do this? Well, me and many others, we look towards electrical circuits. So this is a simple electrical circuit, and you can see the LED does not turn on unless both of these switches are on the on position. So now they're both on, the LED turns on. Now, again, you switch one switch on, does not turn on. This is what people refer to as an AND gate. So you need input A and input B both turned on to get the output turning on. Now, this is a relatively simple electrical switch you can wire together. So I built this myself from parts I bought in the Perth city centre. So, this was really easy to put together, even for somebody with a very rudimentary knowledge of electrical circuits. However, I can promise you, trying to build a circuit into biology is hard. For physics, we have, the constant laws of physics. Wherever you are in the universe, we think these apply equally. But when you're trying to build it in biology, suddenly the rules of the cell are all that matter. The laws of physics are there, but the local cellular context is there. And so, biology is the messy, slimy science for a reason. Things are very difficult. There's all sorts of context dependencies that crop up when you're working with biology. And I hope that through this talk, we'll get to examine this in more detail. Now, beyond the AND gate, so here's a Venn diagram showing what the AND gate would look like. And so green is where it is turned on. So just one input alone is not enough to turn it on, but both inputs, yep, it's on now. Now, this is what we call the truth table, which shows the same information as the Venn diagram, but with numbers, 0 for off, 1 for on. This is the symbol for an AND gate. An OR gate is a related gate, but either input can turn it on. So input A or input B can turn it on. The Venn diagram here is quite simple because it's just all green. Either input can turn it on. Now, a NOR gate is a not OR. So it inverts the OR gate. And so by default, it is turned on, and any input turns it off. Now, again, why am I talking about these mathematical concepts that are very common in making electrical circuits? This is because, to me, this looks like how you might design something in biology. So a NOR gate, to me, looks like a promoter that is by default on, but two different repressors can combine it and turn it off. So suddenly, this is it in the context of biology. Well, people may say, well, biology doesn't really work that way. Well, when I look at the lac operon and how that is regulated. So the lac operon was the first characterized example of gene regulation and won the Nobel Prize back in the 1960s for, pioneering our understanding of how genes are turned on and off. Now, for those who might not be as familiar with it as others, the way this circuit works is lactose turns on this operon in bacteria. Glucose turns it off. Basically, if you can be eating glucose, why bother with lactose? But if lactose is all the food that's available, let's get eating. So to me, the Venn diagram for this looks like that. And to me, it looks like an A implied B gate. And this more or less looks like a particular gate that you might design in a lab, build with an electrical circuit. But this is a biological circuit, a natural biological circuit. And so the rule that, the way we can view natural gene expression looks a lot like some of the electrical circuits and mathematical concepts we've been talking about. So maybe it's not ludicrous to try and build those in. Now, if we were to try and rewire this, though, I would point out that this in isolation does look like a regular, gate for an A implied B logic. But suddenly, if you look at it wired into the rest of the regulatory transcription factors, suddenly it's a mess. So here you can see, oh, here's the LACI, here's the part, the transcription factor that controls the lactose element. Well, here is the glucose controlling element. And you can see how many other genes this is wired into. This is a master regulator. So suddenly, if you try to tweak this one circuit to change how it responds to glucose, suddenly you could accidentally disrupt half the metabolism of the bacterial cell. That's no good for anybody. So what we don't want are these pleiotropic cell-wide effects. So building up a gene circuit from parts not from the host organism, putting them into something, my choice is a plant, then suddenly you're able to, build into new functions without disrupting the existing functions. And so when we put all of this together, you could build a gene circuit, a synthetic gene circuit. And so the logic gates I was talking about really make up this middle part, these processes or the integrated component. So you can take multiple signals and channel them into these logic gates I was talking about. Before you get to that stage, though, you have these inputs. You have a sensor unit. So you have often a protein that will sense, light. It could be specific wavelength of light. Or it could be binding to a particular chemical or ions in the cell. It could be temperature sensing. It could be all these different signals. It could be the location of the plant, what tissue, leaf versus root. And those sensors will then feed into the integrators, integrate multiple signals into each other. That's the point of a integrator, these logic gates. And then you'll get the output. Now, many times in the lab, we're just working with reporter genes. So GFP, luciferase, just to see, is it turning on, off in an easy measurable way to measure if it's working as predicted. But you could make therapeutics or small molecules, metabolites that are of high value. You could change the shape of the plant form, the structure. And so this is how we get into thinking about, well, how can we engineer crops for improved, yield of particular, foodstuffs. And to me, if we can get all of these things working together in harmony, we can make an orthogonal circuit that does not interfere with the natural host machinery, but works independently. Now, I use the word orthogonal. Now, orthogonality is a concept in synthetic biology where you might have two separate signals and you don't want them interfering with each other. So you want, particular output, gene one and gene two, but you don't want the signal for gene one to accidentally turn on gene two. One of these could be natural, the other could be, a synthetic pathway you're adding in, but you don't want crosstalk between, your different synthetic components or your synthetic components in the natural cell. So this is, to me, a very important part of working in synthetic biology, trying to avoid all this messing around with the host machinery. And doing it accidentally when you think you're being orthogonal has been a great challenge. So something to watch out for anybody, starting to work in synthetic biology. Now, why is working with plants so hard? So before I get into some of the data, I just want to really point out that, synthetic biologists, they really love this design, build, test, learn cycle. And many of you may have come across this concept. So you design a gene circuit, you build the gene circuit, so do all the DNA assembly, you're testing it in plants, and you get the result and then you learn from that and you get a very sad looking person because, their experiment probably failed and they've got to go around this loop many times to test it. But one of the challenges plant biologists face is this test step will take a very long time because many plants are very hard to work with. It's very hard to actually transform them, grow up the whole plant and analyze the results. And some plants, we just don't have good techniques for transforming. So, even if we could transform them, it may take years, but in some cases we just can't. I always think like, working with many lab plants like Arabidopsis or moss, they're relatively short cycles, but they're still longer than working with mammalian cells or yeast cells or bacterial cells. I don't want to work with a tree. Working with a tree sounds, terrible because that's going to take a very long time if you're waiting for that tree to grow up and you've got to go through multiple generations. So the length of testing, I think, is a big challenge in plants as well as the transformation aspects. But there are ways to shortcut this and reduce the testing phase. For example, testing in transient assays where, for example, you're infiltrating a leaf of a plant and looking for expression there. Or my favorite is to extract protoplasts, so isolated plant cells, transform them. So this is how we're able to, try to speed this up. But sometimes you've got to go through testing the transgenic plant, getting a whole stable plant, which is going to take a very long time. And then, before getting into the design of the circuits I've been working with, I just want to talk about, well, what about, these circuits? Often you think of electrical circuits, they're digital, binary on off. Does this have any reality in plants? Well, yeah, I think so. So this is an example of a natural epigenetic switch. And so this is a plant where, you've got a mutation disabling the repressing of flowering. But normally this plant will only flower after winter. So it doesn't want to flower in the middle of winter, it wants to wait till spring. And the way this happens, so there's this amazing pathway. So Caroline Dean in the UK has done a lot of work characterizing this with others. And so in normal warm conditions, you've got this 4-OR repressor being expressed. But then during cold, wet weather, it starts to be turned off and an antisense transcript is produced. Then you get this nucleation of an epigenetic mark to keep this gene turned off. And once you return to the warmer weather, actually you get stable silencing and full epigenetic lock in that locus. So you turn off the repressor of flowering, and so suddenly you're able to start flowering and recognize the time of year. So this is just an example where a binary for every allele, every copy of this gene in the plant that has a binary state of off and on, and the accumulation of these being switched off over the entire plant is what leads to the derepression of flowering. And so when we think of digital switches, maybe it's not so far-fetched that we can use them in plants in ways to benefit them. Now, trying to recreate an epigenetic system like this or a DNA methylation-based system, that's a huge challenge. There's a lot of things about the epigenetic system that we don't understand. So being able to write them and target them to particular loci and recreate that, huge challenge. Hopefully one we can overcome. But in the short term, I thought, well, let's take an old-fashioned approach to making permanent changes to DNA or semi-permanent changes to DNA. And in this case, I wanted to look at site-specific DNA recombinases. So in this case, we might have these short DNA sequences that are target sequences for a DNA recombinase. So in this triangle here, you'll have the FRT site. You have two of them facing the same direction. You express the recombinase that binds to them, in this case, FLP or the flipase. FLP will bind to them and it will cut out this region. So the red sequence has been cut out, and you've got left here the single recombination target site. So it is not a scaleless system. You do have a recombination target site left, but this is a very efficient, useful system. Now, these target sites for these types of recombinases usually about 35 base pairs long. So not terribly long, not tiny, longer than a restriction enzyme target site, but rare enough that they're not likely to appear in many genomes too often to cause side effects. So we took this technology and wanted to build it up to make a gene circuit. And the simplest gene circuit would be a yes gate, a simple one input switch. So what did we need to be able to build this? So we needed a constitutive promoter to always turn the gene of interest on. So a reporter like GFPL luciferase, but we needed to, by default, to be turned off. So we needed some sort of repressor sequence, so a terminator transcription makes the most sense. We surround this by recombination target sites. We then, on demand, with a conditional promoter, express the recombinase gene, cut out this terminator, we go from off to on. And one of the advantages of this design is it's a memory circuit. So this is useful if you have a very short signal, like a heat shock or a particular burst of a particular wavelength of light, that you want to translate into a permanent change. So you may not want to continuously heat stress your plant. So a short burst of heat stress can then switch it to a permanent on state. So this is how, why we are happy with a non-reversible system, because there are usefulness of being able to permanently change the switch, have a memory switch. So this was the plan. Planning is easy. Demonstrating is hard. So the approach we took to do this was using Arabidopsis protoplasts. And I would just really like to thank Adil Khan, who works here at the University of Western Australia as well. Somebody you should probably reach out to because he's a fantastic person to get in touch with and work with. So a master of synthetic biology, plant gene circuits, and transforming protoplasts. And so in this case, what we did was, we took the protoplasts, transfected them with PEG and our plasmid, and we were using a dual luciferase assay. So we have Brinilla luciferase and Firefly luciferase. We do this in a 96-well plate, and so we have many replicates of each condition we want to test. We take that to a plate reader, add the different luciferase reagents, look at the ratio. So for example, Firefly luciferase is our normalizer. So it's always got the same promoter. So its expression should be the same no matter what are the changes we make to the plasmid. However, we may change whether or not the renewal luciferase is turned on or off based on, whether or not a recombinase is present, varying whether that promoter here can be turned on. So this is a basic schematic, and we use the 96-well plate, so we're able to look, at many different plasmids at the same time with replicates. So this is where I want to introduce a concept called, hard-coded circuits. So when we think about our gene circuits, I think of it, the actuator, we can think of that as the output. Now, a zero input circuit state would be the actuator is just there without any inputs. So there's no recombinases. If we were talking about my recombinase circuits, there's no recombinases present. If this is a NOR gate, its default will be on. We take this plasmid that doesn't have any inputs, doesn't have any recombinase genes. We transfect it into a population of cells. And then we compare that population of cells to another population that does have one of the input genes, so a recombinase gene, for example. We then have input B, and then eventually both inputs present in the same plasmid. And we're comparing between populations of cells. So we've hard-coded whether or not the circuit should be on or off into the plasmid. So we transfected that into the different populations of cells and compared them. This is in contrast to a soft-coded or dynamic circuit where you have a single plasmid and you have all the inputs present there, but each input is controlled by a different, condition. So again, heat shock, temperature, developmental stage, they can all turn that on. So you'll have different plants that are exposed to different treatments. So they're all genetically identical plants, but then you compare, you might treat one with heat and one without heat, and suddenly you'll see the difference of your circuit. So I wanted to outline this because we're going to be talking about both throughout the rest of this talk. So now to build up the memory circuits, trying to build that yes gate, we tried to put this together. And so we have a range of different constructs we built here. And you can see this is our control plasmid. So whenever you see blue on the graph, it's based on this plasmid here. So this is the actin-2 promoter driving renylluciferase. There's nothing blocking it, nothing tampering with its expression. And then I built a series of different plasmids along with Patrick, who was an undergraduate in the lab. Now he's doing his PhD on gene circuits. And so together we built and tested a range of these different repressors to see which one works the best, which one turns off the expression, because we need to turn it off before we turn it back on. And you can see here this short TB transcription block synthetic terminator. Didn't really repress well. It did repress statistically significantly, but not to the point where I would consider that off. The OCS terminator and any version of that, quite good repression. We were hard to get repression below this point. So this was sort of like our baseline for off. Surprisingly, the 35S terminator from the cauliflower mosaic virus didn't repress at all. And this really confused us at first. We thought, well, wait a minute, we use this terminator all the time for our constructs. We should be worried about this. So Patrick did some reading and he found a paper from, I think it was 35 years ago, where they had actually tested different placements of the 35S terminator relative to the promoter. And if it was really close to the promoter, did not act as a terminator at all. And so actually we just rediscovered some old biology through this experiment. So this was wonderful to remind us, like, not all biological components are interchangeable Lego blocks. I love Golden Gate cloning. This is what all of this was built with. I think this is a great system, but I think it's really important to remember, just because a part is labeled as a terminator part in your favorite Golden Gate kit does not mean it's a terminator that acts the same as all the other terminators. They're not purely interchangeable. Each one has its own behavior, dynamics, effects, and they need to be well characterized in your host organism of choice. And in this case, we found that the distance between the promoter and the terminator made a big difference for this particular terminator from the 35S. But it didn't make a difference for the OCS terminator. So again, not all terminators are interchangeable. I also tried an upstream open reading frame that's supposed to induce RNA decay and translational innovation, but sadly we only saw a modest suppression. So moved on from that. That sort of was inspired by some of my PhD work, but it was like, did not help with the synthetic biology world. So then we wanted, can we turn it back on? And again, this is the hard-coded circuit. So we were using constitutive promoters to always turn on the recombinase gene. And we found that all of the tested promoters, whether it was NOS, TCTP, actin-2, 35S, they all turned it on. But the one that gave us the highest activation was the TCTP promoter. Interestingly, this has a similar expression level to actin-2 and is lower expression to these two other promoters. So it's not just pure expression level. So there's clearly other factors affecting, what led to the best activation of the circuit beyond just the promoter strength. So, there's a lot of interesting biology there. It could relate to stability of the mRNA, propensity to silence in our protoplast system. But, we found that this was a really good set of promoters to work for our circuits. So then I transformed this, made a stable plant where the circuit was heat inducible. So this is going back to that dynamic circuit idea. So this was hard coded. Can we see it dynamically change from off to on in genetically similar plants? So here, instead of using luciferase, we were using GFP that was nuclear localized. And we used heat induction for the FLP recombinase. And you can see here, these plants here, not heat shocked, and we can't see any GFP in the nuclei. But as soon as we take the sister plants and heat shock them, suddenly you can see all these nuclei lighting up. It's like a beautiful Christmas tree. We're seeing a lot of GFP being activated. So, here, this shows that we're able to build these circuits and they're able to function in stably transformed plants. So after the confidence I got from working on all of this, I thought, oh, easy. I'll make an OR gate. I'll make a 2-input switch. It'll be super simple. No problem. I'll just take Cree, the most commonly used recombinase in all of plant biotechnology. I've already got Cree working, put them together, OR gate, no problem. It failed. So, this was an important lesson, another important lesson for synthetic biology. You've got to test everything in isolation before you start putting them together, because then you don't know what's causing the problems, what's causing the headaches. So this circuit design here is the OR gate. The terminator is surrounded by target sites for the 2 different recombinases. So if you express just FLP, it cuts out the terminator. If you express just Cree, it cuts out the terminator. If you express both, it will cut out the terminator. And we saw it's off at FLP, cuts out the terminator. If you add Cree, well, might be cutting out the terminator, but it's not turning on. And if you add both, suddenly Cree stops FLP from being able to work. This plasmid and this plasmid are almost identical, except this has a gene encoding Cree. I thought of many different reasons why this could be a problem. It's taken us years to actually solve this problem. Hopefully we'll be able to publish that soon. But like this was so frustrating. And again, this is the most commonly used DNA recombinase in all of biotechnology. So it failed me, it betrayed me. So, okay, I went hunting for another recombinase. And so at the time, Florence, who was an undergraduate and honors student in the lab and is now also a PhD student working with us on gene circuits. So she started screening, well, let's find another recombinase that can work. And so she screened S-Cree, V-Cree, and Vicar, all relatives of Cree, all of them equally disappointing to me. Only V-Cree had even a modest increase in expression, and that was not impressive enough to try to build new circuits out of. But we were also able to test a relative of FLP, and FLP, it was able to activate. This B3 recombinase caused, great activation. And so B3 had come here to save me. And I was very happy, but I had learned to not trust anything. So I thought, well, what happens if B3 can bind to the FRT sites or FLP can bind to the B3's target sites? What happens if they can cross-react with each other? It's been published in bacteria and animals that they don't cross-react, but maybe in plants it's different, because now I just don't trust anything. And I tested it with Florence. We tested it, looked for cross-reactivity. No cross-reactivity. They're just off. So this is amazing. Wonderful. So finally, we can go back to building the 2-input circuits. And we did, and they worked. So we built an OR gate, and this time B3 or FOP targeting. And we saw, yep, activation. We turn on in all of these conditions, whether 1 recombinase or 2, we see activation. Now I built an AND gate, an even more complex repetitive plasmid. It has all the same genetic parts as the OR gate, except we have two terminators, each one separately surrounded by recombinase target sites. So to turn this on, you need to cut out both. And again, we see from the data, you need to cut out both before you get activation. So you get no significant activation until both recombinase genes are on the same plasmid in the same cell. So this was a huge triumph. We were able to build, 2-input logic gates behaving themselves as predicted with these recombinase components. But really what we care about is not protoplasts, but actual plants, stably transformed plants. So I spent a long time building up designs to show that this works in stably transformed plants. So again, going from these hard-coded circuits in protoplast to this soft-coded dynamic circuit that has a sensor that senses the world around it and responds. And so the sensor we built involved chemical perception, so dexamethasone. It's a common inducer of gene expression in plants. I have a lot of opinions about it and whether it's useful or not. But we managed to get it to work in this system. I also wanted to look for cell type specificity. So this is the cortex of the root, so the second layer of cells within the root. And each one turned on a different recombinase. The output, again, this is nuclear localized GFP. And this time we're using the very strong 35S promoter to make sure we could capture anything we saw. And you can see here this root, nice, lovely root, lots of long root hairs, no GFP at all. But then if you add the chemical dex, suddenly you see lots of GFP nuclei. And if you actually do an optical sectioning of the root to see which cell types it is, which cell types have the GFP signal, it was only the cortex. It was only this second layer of cells. So this confirmed that we were getting induction only in the cortex and only after we added dex. So again, AND gate logic, but at the cellular level right now. This I was very happy with. So again, arrogant, cocky. So I thought, oh, I can build anything. Let's build a repressive system. Let's build a NOR gate. So it's by default on, and then it turns off. I can do anything. No, I couldn't. It failed. So what we had here was we wanted to cut out the CDS of the reporter. We're going back to cells. We're going back to renal luciferase. We tried to cut out this CDS. And after 24 hours, FLP had repressed, but B3, no repression after 24 hours. B3 had been my savior. B3 had saved me when Cree had failed me. And here I am giving personality, motives to proteins that do not care about me or the world around them. So please forgive my anthropomorphizing of proteins and enzymes, but I felt betrayed by B3. But after checking out the 48-hour time point, I saw it did repress, but it was repressing slowly and poorly. So it wasn't as bad as the first case where we just never saw activation due to Cree in that OR gate. In this NOR gate, we were seeing repression, it was just slow. So I sat down and had a thought, well, why is it going slow? What's different? Because it was working very fast in the yes gate where we were going from off to on. Here we're going from on to off. What's changed? FLP is still fine, B3 isn't. I thought maybe it's the distance between the recombination target sites. In the yes gate, the two B3 target sites were 700 base pairs apart. Here, they're over 2 kb apart. Maybe the further away they are, the less efficient B3 is. Couldn't see anything in the literature about B3 and whether this was true, but I thought worth testing, worth trying out. So I thought, well, how else can we turn a gene off other than cutting out the CDS? Let's cut out the promoter. So I decided, well, let's relearn the lesson I thought I learned after Cree and test everything in isolation. Test everything in isolation. So instead of building a NOR gate, let's build a NOT gate, one input. And so we tested these. I won't go into details, but this replicated what we saw with the bad performing B3 action in the NOR gate in the single input NOT gates. But suddenly, if we're cutting out the smaller promoter, which is only 800 base pairs, not over 2 kb, suddenly B3 acts the same as FLP. So we're able to build a repressive circuit with B3, but we have to target the promoter, which is smaller. So it could be a context dependency to do with length and differences between FLP and B3. Again, different enzymes, different genetic parts, promoters, terminators, enzymes, even if they have the same mode of action, they often are not interchangeable parts and they have their own unique behaviors. You could anthropomorphize them, say attitude, behavior is maybe, less charged. And so this is such an important lesson. But now I had learned this lesson, or relearned it, I was able to build an A-implied B gate. So this implied gate is similar to the lac operon I mentioned earlier, and we're able to get induction turning on of a gene when only FLP is present, but if B3 is also present, it turns off because it's a dominant negative. We're able to override that signal. So if we never want it on. I think this is demonstrated a wide range of circuits we're able to build, and it's a real delight to be able to sort of share this. And I just want to, with these circuits parts, I just want to touch on a couple more examples. So, Patrick and Florence, they did a lot of work with what we call a split recombinase. So this is the protein crystal structure of FLP bound to DNA. But if you split the protein in half with known split sites, you're able to get this. You're able to get two inactive halves of the recombinase. And what we did was we fused a homodimerization domain C1 onto each. And so then they will dimerize when expressed in the same cell, reconstitute a functioning enzyme. And you can read more about it in the paper, but we were able to test this and show that we could get an AND gate, a split AND gate working, and a NAND gate. So this is the default on, but when both inputs are present, you turn it off. So this, was fantastic. So this hopefully demonstrates the range of logics we're able to achieve with our memory circuits implants. And I'm looking forward to being able to share a manuscript shortly where we've expanded out, what we can do with programmable logics with memory circuits. I just want to change gears though, because I think this will be very interesting for the audience for this particular talk, because I've shown off a lot of circuits, but underpinning all of that was cloning DNA, building large, often repetitive plasmids using the same promoter elements, same terminator elements over and over again. And sequencing of those plasmids is very difficult. Many people don't sequence their plasmids beyond the first thing they clone. But often when you go to these higher levels, they're level 2 in Golden Gate, terminology, but just large plasmids with multiple different parts on it. They're too large. They cost hundreds of dollars from Sanger sequencing. And Illumina sequencing of the plasmid is challenging, especially because the short reads of Illumina don't reconstitute it very well. So I've been thinking about this problem, and we've been using a lot of Illumina sequencing to analyze internally in the Lister lab to look at this. But again, the challenges of repetitive large plasmids is still there. Now, many people use Plasmidosaurus as a fine service. It seems like a really nice service, but the turnaround time here in Perth, Australia is too long. They're quite pricey, and we're very price sensitive. And Perth is a very isolated city. Our next nearest city, I think, is either Adelaide or Singapore. We are, in the middle, we're surrounded by ocean and desert. And so trying to come up with innovative ways to speed things up locally has really spurred us on. And hopefully, what I'm going to talk about may also be helpful for others. And so I've been playing around with Oxford Nanopore's MinION to sequence plasmids, and this has been, electrifying to me. So if you take your unconfirmed plasmid, even if it's large and repetitive, you use the rapid kit bought from Oxford Nanopore. This uses TN5 enzyme, I believe, to barcode it. So it cuts and barcodes your plasmid. And what you get is a diverse mixture of fragments being produced. So you'll get occasionally the TN5 cutting your plasmid once. And so you'll get a read that represents the full length of your entire plasmid. You'll get some where it's cut and you've got long reads that represent a very large fraction of your plasmid. This is relative to the size of your plasmid. If it's a 3 kb plasmid, your full length reads are going to be 3 kb. But if you've got a 12 kb plasmid, well, your full length reads will be 12 kb. Your long reads could be, 4, 5, 6 kb. So in terms of trying to then assemble this computationally to see if you've got your actual plasmid, that's going to be a lot easier. So sequencing this with a laptop, so this is a drawing of my laptop and my MinION plugged into it. You run it, you're able to convert that into your DNA sequence information. So this is a FASTQ file for people who do any computational biology. And then you can assemble it into a full plasmid, annotate it, compare it to your expected. Given how well this can look at highly complex repetitive plasmids, blows my mind. I think this is amazing. At the moment, the cost is, I'm estimating in my lab, it's around $10 Australian. The Australian dollar is not the strongest currency relative to the US dollar. So, that price is not terrible. I understand it's still high to many labs' operating budgets, but I hope to be able to bring the cost down even more. And if anybody else is doing something similar and has tips, like I'd love to start exchanging ideas. I'm more than happy to share protocol information. I'm hoping to release it all in the near future. But if you want something like the development phase protocol, please reach out to me, email, LinkedIn, Blue Sky. I'm happy to talk about it more. But this is an example of how well it works. So this is one of my favorite plasmids. This is that AND gate from a few slides ago. And you can see we have this OCS terminator, 700 base pairs, perfectly replicated twice near each other on the plasmid. We have a lot of 35S terminators, TCTB promoters scattered throughout it all. And so trying to assemble this with Illumina short reads is, as far as I can tell, impossible. I've never seen it fully assemble from short reads because these repeats are longer than the read length, the fragment length in Illumina sequencing. Here you can see with either 50 nanograms or 20 nanograms of input DNA into the sequencing reaction, we're able to get perfect assemblies with the CanU assembly software. With 5 nanograms, no assembly. So that was too dilute. But even with 20 nanograms, which is below the recommended amount, we're able to get full assemblies. If you wanted to see what the mapping looks like, you can see here the noisy long reads from nanopore, all here, and you can see, that the average pileup, which shows, whether or not you've got a consistent mutation throughout, perfect, base perfect throughout. So you can see why the 5 nanogram didn't assemble. You've got regions with almost no coverage. So, and you've got areas where they you appear to have mutation when you have greater sequence depth, you don't see that mutation. These were the same plasmid from the same tube, just diluted down. So there is definitely an issue where if you're too dilute with your DNA into the sequencing reaction, that's not going to work. But if you have sufficient DNA concentration in the reaction, you're able to get an amazing assembly of even large, complex, repetitive plasmids. To me, this is, a game changer for a lot of the synthetic biology work. It's relatively quick to both make the library and sequence. The data analysis is also quite quick, depending on how powerful your laptop is. So, the turnaround time for this is amazing, and the buying cost is low. Like Illumina machines, the cheapest Illumina machine is going to be maybe 50,000 US dollars. Maybe it's less than that, but you're talking tens of thousands of US dollars. For this is 2,000 to 3,000 US dollars entry fee, which gets you not just the machine but the reagents. So you're basically getting the machine for free in exchange for reagents costs. So the buy-in level, still significant, is so much less than the tens of thousands of dollars it requires to be able to buy Illumina and get into that ecosystem. So the entry point for this ecosystem is significantly less. So I hope to see this democratize, in-house plasmid sequencing across the world in a really fundamental way that can hopefully open up synthetic biology to many more people. So, enough about this is sort of like a side story, but I'm really excited about what this can offer, not just my research, but people research all over the world. And so I'm already coming to my time, so I won't spend too long talking about this last story. But I wanted to say not just memory circuits exist for plants, but there are other gene circuits. So Jen Brophy has led the development of some amazing circuits using bacterial transcription factors. And my colleague Adil Khan, who I mentioned earlier, the wizard at protoplast transfection, he has led the development of CRISPR-I-based gene circuits. And I just want to very quickly give you an overview of those because I think they're fascinating. And if you don't like my memory circuits because they're not reversible, these systems allow for reversibility. So this is a beautiful system using CRISPR interference from Adil. So the idea of a NOR gate is that it's a universal gate. So its default is on, you turn it off with either input. Because it's a universal gate, it means you can layer, connect NOR gates together to achieve any logic. The computer that went to the moon on Apollo 11 had around 5,000 NOR gates on it, and that was it. Much less than what's in most people's laptops or phones, but enough to do the calculations there. But it was all done through NOR gates, layered NOR gates talking to each other. So to build a single NOR gate, what Adil did was he engineered a promoter. So he took a common plant promoter. He actually took a couple, TCTP and 35S, and he engineered them. So he found their Tata box, because these promoters do have Tata boxes. Not all do, but these ones do. And he engineered strong binding sites to a guide RNA around them. So rather than making guide RNAs that bind these natural promoters, he engineered the promoters to bind strong guide RNAs. This is a problem that I think many people didn't appreciate. You think, oh, CRISPR, the guide RNA can bind to any sequence. No, they don't bind to all sequence equally well. So he reprogrammed the promoter to recruit DCAS9 efficiently. So I think this was the special, innovation that he really brought to this. And so this engineered promoter, the integrated promoter, here you can see him demonstrating, oh, it is acting as a NOR gate. So it's on by default, either input is able to turn it off. This is in isolated protoplasts. He then took it and expanded it out to make an OR and an A imply B gate. He's also made an AND gate, but you'll have to read the paper to see that. He's also then tested it in stable transgenic plants to show what works, what doesn't work. So you can see here he's having problems. DEX does not induce repression, but heat does. And he's able to show how this is reversible and how this comes back on. And he's also tested it in a range of different plants. And while we see it needs optimization to get it to work in some species of plants, there is promise of this system being fairly universal, and with some optimizations, it can work in other plants. Here's from the review article that we wrote earlier this year, where we compared different gene circuit systems. So if you want a comparison of these different systems I've been talking about, you can check out the review article. It is open access, so but if you have any issues finding it, please reach out. And so really, I think we've built a lot of amazing technologies. There's a lot of promise of how we apply the technology to these different applications, whether it's about making stress-resistant plants, pathogen-resistant plants, or making new chemicals, but moving it from host to host. So I've talked a lot about working in Arabidopsis and how challenging it is when you change one genetic element, suddenly things go wrong. Imagine going between banana and strawberry, very distantly related plants, how many challenges you're going to have, how many plants that are important to people that we cannot transform, that we cannot introduce new genes to, that we can't even regenerate and do CRISPR editing on. So this plant transformation bottleneck is definitely a huge challenge. And I don't want to take up too much time, but there's one final story, because I know you're interested in genome sequencing, and this relates to genome annotation. So this is new work from my lab this year. And so Moj here has helped lead a lot of this. And so he's been a visiting scholar working on this annotation project, as well as some SynBio work. And so this is the Arabidopsis reference gene, annotations, gene models for a particular gene, which is an RNA splicing factor. This links back to my old RNA decay and splicing days of my PhD. So less synthetic biology, but the genome annotation hopefully will be interesting. So this top one here, you can see the start codon goes through, stop codon here, normal looking gene. Suddenly here, you've got a splicing event, and this introduces an early stop codon. However, the Araport 11 reference shows, oh no, the start codon just is late, and the stop codon's normal. But in reality, biology doesn't care what the longest open reading frame is. It will still start translating here, and it will find a stop codon around here. Here, the annotation software is just finding the longest open reading frame. And so you're getting this sort of protein that's wrongly truncated at the beginning. We use some software that was developed by a group in Scotland, where they do annotate this appropriate start codon throughout. So this authentic start codon. So now we find the real stop codon, and we're able to see how long and unusual this 3' UTR is. And this relates to RNA decay mechanisms. I won't go into details. And you've got more details here, but I'll skip over this in the nature of time. And just here, I think it's interesting from a protein crystal structure point of view. So you've got the full length protein here. And if you look at the reference off, you see, well, it's still a fairly long protein is predicted to be made. But if you look at our revised one, which is more likely to be a product if it was translated, suddenly it's tiny, it's truncated. So I think wrongly annotating the start codons in your transcriptome can have big effects on your interpretation of alternatively spliced genes. So if you want to read more about this, please do check out the preprint. And we'd love to hear feedback as we're planning to submit it to a journal soon. Um, and I'd also just like to advertise, and please share this widely. We are recruiting a postdoc to join us on a new project. So I'm really excited to be working with Ryan Lister on a new collaborative project funded by a UK funder called ARIA. And so they want to build the first synthetic chromosome in plants. So how can we go from just transforming plasmids into plants to building something up that's a linear chromosome with centromeres, telomeres, all those wonderful things, get it to a large size, and how can we get it to function in diverse plants? And we'll be working with some moss, and I'm really sad I didn't get a chance to talk about moss, my favorite plant, more in this talk. But we also then want to get it into potato and eventually other crops. And so this is an amazing undertaking in terms of like engineering at scale. And so this is where the genome rewriting really comes from. So building gene circuits, we're rewriting small parts of the genome to add new ways in which we can turn genes on and off. But here we hope to be able to build whole new chromosomes and move them between plants to add new functions. So I'm really excited for what this project will bring. You can see more about the job posting. We're looking for a person with a lot of experience with microscopy of chromosomes so that they can view whether or not the synthetic chromosome we're building is actually behaving as expected. So please reach out to me if you're interested. Posts are on LinkedIn about it, so you can check out the job posting there. And with that, I've got a lot of people I'd love to thank: the Australian Research Council, ARIA for funding, and also the University of Western Australia, the fund that's funding my current position, my fellowship. Deeply grateful for this opportunity, and yeah, I'm happy to take questions.
Thanks a lot, James. That's wonderful. Yeah, so for those who may have questions, please feel free to type in the chat or just raise your hand and we'll call on you to speak up. So just before we get into the participants' question, I think one thing I want to clarify with you from the talk. So you show your experience with the logic gate building in plants, in which you started from the FLP FRT system, and then you just moved to the Krelok system, which is also another widely used system and it has been very successful in yeast and in other organisms. It just failed miserably. Do you have any clue what was the failure mode? Is it because it doesn't express? Is it because the enzyme is bad, or is it because like it doesn't get to the DNA?
So, I'm trying to think how much I can say without ruining upcoming manuscripts and the secrecy that the Lister Lab has over some things.
Yeah.
So we think it's recombining. We think it's cutting out the terminator, but we
Hmm.
think it's sticking to the DNA too strongly, and so it's interfering with other things. Oh, that's part of the problem. But I
Yeah.
think what surprised us was when we solved that, we found another problem as well. And so this is again relates to the context dependency of these genetic parts. You put some genetic part next to a promoter, and you think, oh, there's not going to be a problem here. Why would this interact with that promoter? Why would there be some sort of mechanism connecting them? There shouldn't be. It turns out that that's more common than you think. So the Cree actually there is a couple of different problems. So hopefully that's not too disappointing an answer.
Yeah, yeah. So it's like maybe like the clear, the short answer would be like context make a lot of difference when you move from organism to organism. When you move from a plasmid to a genome, then everything is changed.
Mm,
Yeah,
absolutely.
yeah. Well, if there's any questions, please feel free to put into the chat. Yeah, I do have another another question about the nanopore thing. So,
Mm.
so I think maybe like this is like more of a sharing, but like, um, have you been having, some success by changing the concentration of the reaction, blah, blah, blah. So I believe like I haven't done the actual work myself, but um, I see a lot of concerns where like if you're doing the reads with the amount of you added in, sometimes you can multiplex them and you can add more, and you don't need a lot of reads. You don't need a lot of good quality reads in order to assemble a whole plasmid. So in that case, when you improve the standardization further, you can actually lower the cost by a lot. And I think that's like what the company around here are trying to do as well to, push it further, including maybe like Saras and like Quintara Bioscience here based in Boston and San Francisco is trying to do something new. Some of them get the price as low as $5 per whole plasmid sequencing already. So that's pretty interesting on the technology that's coming in.
And I think it'll be hard for myself to try and compare to that, especially with high salary costs. So like when I'm quoting those numbers, that's purely from a reagent point of view. I'm not taking into account the time it takes somebody to prep it. So at the moment, those numbers were from multiplexing 24 plasmids together. There is a version of the kit for 96 plasmids that can be multiplexed together. I'm waiting for that to arrive. I'm hoping to get that soon and experiment with that. So I think multiplexing 96 together will hopefully help bring the cost down to sequence more, plasmids per flow cell per run. Luckily with Nanopore you can reuse the flow cells, but I think getting more plasmids into each one of those runs may also help. But we'll see if that actually is true when we get experimenting with it. In terms of changing reaction volumes, I'm actually using what the kit recommends. So if I was to miniaturize the reaction, that could save money significantly as well, because at the moment library cost is very significant. Library prep, the kit is very expensive. So my next step is to try and increase the number of plasmids per flow cell and bring the cost on the sequencing side down. And once that comes down, then I'll start thinking about miniaturizing the reaction more. But again, if other people have already, had experiences with that and they're able to share their findings from that, that would be very useful to help, us, figure out what the next step is. But already it's proving a very valuable tool for us to be able to work with in the lab and speed up, our throughput for cloning and validation.
Yeah, that's very true. And I think like maybe another factor to be aware of is the concentration of each plasmid.
Mm, mm,
Because
mm.
like if you have too much of one over the other, then the abundance of the readout will just be skewed towards those one. I think that's one of the key important thing that the workflow here in the commercial labs are trying to do, because they try to make sure that they're not the one normalize these things, so they have to work with the sample that come from
Mm,
everyone,
mhm.
like from different labs to work on that. So that might be a good thing to touch on. Like, unfortunately, these are mainly done at the company, so I don't see a lot of them
Mhm,
coming out
mhm.
in publications. So they just
Yeah,
consider
yeah.
that as a trade secret, or maybe just like, they just didn't spend that much time trying to put it out because it's a very fast growing field. Yeah.
Yeah, and that's something I really want to change because lots of people, both labs and companies, are already doing this, and I've not seen much in terms of publication. And I would love to see publications making it more accessible, advertising it, showing how you can actually do it, where the hurdles are. Because without somebody telling me about it, so a colleague at Curtin University here in Perth told me about his experiences with it, so I learned from him, but I don't want it to spread by just purely word of mouth, like, oh, the lab or the university next door is doing it. No, I want to really share protocols, share information more widely. I'm sure there are people already doing this to some extent, but like the more of that, the better. The more we get the word out, because I think like having in-house sequencing capabilities, gives you a lot of power and speed over these things, having control over it. If Plasmidsaurus or similar services are working for you and the time and cost is fine, great. But for me, having the power to do it in-house, control the throughput, control the timing, bring the cost down and, do it on a schedule that makes sense for me, especially given the long shipment time and wait time for even Plasmodsaurus, one of the great companies in the area, which, I have great respect for them, but Perth is an isolated city. So the turnaround time, it's always going to be hard unless you've got a facility in the city doing it. And Perth is too small a city to probably justify it right now. So doing it ourselves as a local service where we don't care if we're maybe losing money, from charging other colleagues doing it, because, being able to do it, share, help each other out is more important.
Yeah, thanks a lot, James. See, that's a very important thing to be, to have like the control of what is going on in the lab, and that's true for the place that is like maybe like further away from the big hub, because like the big hub is usually where like the fast growing companies were, and they were like the competition sort of like help things move a lot faster. So we have a question in the chat from Christine, and Christine is actually from the IGM team in Uganda right now. The team called SynBio4ALL is like trying to do a lot of stuff, and that includes like the engineering the algae, which is like also bacteria, which is like having some relevant to the plant. So the question is kind of long and have like a lot of points to it. So Christine, if you can unmute and like say it out loud, that could be very helpful. So were you able to unmute, Christine? See you unmute. And they're trying to make tyrosines in the microbe, so that could be something. And sorry for the background noise. I think someone's starting to kill the nature around my house right now.
Glucose in high concentrations influence the synthesis of other cellular nutrients, and also like cellular nutrients, like maybe the amino acids, and also can its availability suppress or enhance the expression of pathways involved in the biosynthesis of other essential metabolites. Then also, should I first stop and you respond? Because questions are many.
So I think I missed the beginning of your question. Can you say the beginning part again?
Does the production of glucose in higher concentrations influence the synthesis of other cellular nutrients in the cell? Since you said, okay, it's okay.
So I think I got the question, which was, does glucose then interfere with downstream metabolic processing of different pathways, different amino acids? Is that what you're asking about broadly?
Yes.
So I'm going to admit I'm not a metabolic engineer. Those metabolic pathways, that diagrams that some people draw, are very daunting to me. But there's absolutely effects of what you choose as the carbon source for an organism. This is going to have major impacts on the downstream metabolism. I remember seeing work in plants, so not cyanobacteria, but where you would give different carbon sources. So, not just glucose, but maybe an amino acid, which also could be broken down into, energy. You give it certain amino acids, you suddenly change the metabolism of the plant, the leaf tissue that's being analyzed. And you're having all these unintended consequences on, the particular amino acid you're trying to give it as an energy supply, and suddenly it's inhibiting particular pathways. And so suddenly you reveal, oh, this particular amino acid affects translation rates, or, because overproduction of this amino acid has all these negative impacts. So, the choice of carbon source, how you're affecting flux through different metabolic pathways, they definitely have all sorts of unintended consequences. Did that get to that part of your question?
Yes, it did. Then the next part of the question is, does that availability suppress or enhance the pathways involved in biosynthesis? I guess that's an also hinted.
So are you after, like, can you at the genetic level, sort of regulate this to try and offset those side effects?
Yes, please.
So I think in many times, yes. So I think it really depends on the particular like effect. But I think you could create, sort of gene circuits or different regulators, artificial transcription factors that could control particular fluxes through the pathway, and you could time it. So I've seen people try to build timer circuits in bacteria where you turn on one enzyme in the step, and then the next one will turn on at a predictable amount of time after the first one, and that allows for the flux to pass in a particular way. So there's definitely ways to time that and regulate it in different sort of circuit ideas. I think in the plant world, we're still very new to gene circuits. We're decades, literally decades behind some of the other fields, especially bacterial work. So they're getting very creative with the circuits they're designing in bacteria. But I think there's some amazing ideas out there for, like, timing circuits to control a metabolic pathway. But there's also other ways you could control this. So I think subcellular localization is really important. So in a eukaryotic cell, you've got many different organelles. So the chloroplast and mitochondria, as well as the peroxisome. And so, concentrating biochemical pathway in one of these subcellular compartments could be important. And what I think is really exciting is the production of synthetic organelles or designer organelles. So there's a beautiful study from 2017, colleagues of mine at the University of Leeds, they had built a designer peroxisome. And in theory, you could develop this, and this was done in a moss. And in theory, you could do this and concentrate a biochemical pathway in there and protect the product of that biochemical pathway in that part of the cell. So it's not interfering, it's not inhibiting other reactions outside of that subcellular compartment. So you could maybe make something like a membraneless organelle with RNAs or proteins with intrinsically disordered regions in a bacterial cell, for example, and concentrate metabolites or enzymes within there and have that maybe have less of a profound effect across the whole cell. At this point, I'm more speculating, but that could be an effective approach.
Thank you very much.
And,
Still have lots
thank
of
you.
Thank you for question as well, Christine. I think that's really great to hear from your interest as well. If there's any other question, please feel free to type into the chat. Do you also have another question, Christine? If not, then I may give the floor to you.
I do have another question.
Oh yeah, please.
This one is related to. It is related to. It is related to. It is related to genomic writing, and with the increasing advancement in genome synthesis and editing technologies, we often hear about the writing and the minimal or optimized genomes in microbes. But considering the complexity of the large size of the genome, what are the major technical or biological barriers to achieving full genome writing plants, especially in terms controlling epigenomic regulation, polyploidy, and then the non-coding region? Also, is there a foreseeable pathway where we might design plant genomes from scratch for desired traits like drought or resistance, then the high nutrient yield? Or can we still be limited to editing rather than full writing in the near future?
So thank you. That is something I've been thinking a lot about with this new synthetic chromosome project. How do we build large, complex DNA for a eukaryotic cell? And there's many limitations here. So one simple limitation is if you build a piece of DNA that's over 100 kb, it's going to just fragment and shear. Like, you build it in vitro, even if you could build something that large, which is already a challenge, if you getting enough of it is a challenge, so amplifying it, then it's not very stable, it will shear. One of the limitations for nanopore sequencing, DNA sequencing, is not how long it can sequence for, but how long your DNA fragments are. Getting a megabase long piece of DNA intact from a DNA extraction is very challenging, very rare, even for high quality DNA prep. So the shearing of long pieces of DNA when they're not in the cell wrapped up in chromatin is a big challenge. So already we've got the challenge of, well, how do you deal with large synthetic DNA? So, I'm saying over 100 kb is a problem. Plant genomes, the smaller plant genomes are over 100 megabases. So, many orders of magnitude more. So from a synthesis to plant point of view, how do you get large synthetic genomes made for a plant that are on the many megabase scale? And then how do you deliver them? That is a challenge we don't technologically have a solution to right now, and is something that's part of our project. How are we going to try and innovate and overcome that? How do we start small, get bigger? How do we do that in an organism? So, that's a physical limitation. How do we deliver that much DNA? How do we keep it stable? Another limitation is the design. Like, how do you design it? Like, there's not really much software for designing genomes from nothing and making a minimal genome. There isn't really much software that I'm aware of, at least for eukaryotes, to do that. So this is a big challenge. How do we understand the non-coding? How do we understand the coding? Like, these are all problems that hopefully by building genomes, trying to build minimal plant genomes, we can learn. So not only can we make a streamlined genome that could be beneficial as a product, but through the process of making it, we'll break lots of things and figure out more about the basic biology. So I think this is a really exciting field. I'm really, enjoying entering into this. Just hiring my first postdoc at the moment to join working on this project, and yeah, it's an absolute blast. So hopefully that answered a lot of your questions about the genome synthesis.
All right. Is there any other question? If not, maybe like for the interest of time, I wanna thanks James for, walking us through a lot of interesting topics today. I think like another thing that I found from, like listening to your talk is that reminded me to like my colleagues at U of Washington, where they also study plants in bio, and they were working on integrates instead. I put the paper there in the chat, and that was pretty interesting approach when they're trying to do like the plant root development, because they're trying to use the plant signal instead to see what is going on. I think that was another interesting thing. And I think like Sarah Cuscio, who is now at the UK, having her lab there, should have like some other works coming in soon as well. Yeah.
So I've not actually had a chance to meet Sarah yet, but her work, the Nature Communications paper, was such an elegant piece of work where, really detailed examination of how these genetic components work, how you can fine tune them, and how important the sensor, the developmental switch is. And so using these hard coded circuits I kept talking about, that's building circuits in easy mode because you're not having to deal with the sensor component, and any leak in the sensor is such a big thing. So, huge respect for Sarah because she took on a really ambitious, set of challenges to directly work in development and be able to work through that. So, yeah, that's a must read paper for anybody, joining my group.
Yeah, that's pretty cool. Well, if there's no other question, maybe like this is a good time to wrap things up. Thanks everyone again for joining us today, and there'll be a lot more people who will be coming in to check in the recording. And like for the last, thanks James for walking us through the very interesting work. We will help and take part in helping the spreading the words about your new lab as well, just so that like a lot of great people are interested to joining your team and move to Australia and explore different type of plants there.
Just to sell Perth, amazing city, really love it. No, it's been a real pleasure. It's nearly 10:30 p.m. here, so yeah, I should probably get off to bed and get ready for tomorrow's early morning lab meeting.
Thanks a lot, James. Thanks a lot for staying late for us. Well, with
No,
that,
happy to.
yep. All right, let's stop the recording now.